So today we will discuss about antibodies, right? Basically, antibodies are the glycoprotein. They are glycoprotein, but they have high amount of protein in it. High amount of protein. Basically, they are soluble in our body. They are soluble in our body, and they are present. They are basically present in blood and lymph. Right? They are present in blood and lymph. So now let's talk about the structure of this antibodies. Right? Let's talk about the structure of antibodies. Basically, this antibody have two chains. Let me discuss about the. Okay, basically, this is the structure of a antibody. This is antibody. Basically, the antibody comprised of two chains, two types of chains. For example, we have one is basically there are four chains. Two are heavy chains. Like I can't write this. There are four chains. There are two heavy chains. And there are two light chains. Right? Basically, this chain and this chain, these are heavy. They are heavy, and they are light chain. They are light chains. Right? So basically, the heavy chains and these light chains are identical. These both are identical, and these both are also identical. Basically, there are two sites in these antibody. We have two sites. One is known as the binding site. Basically, first is our variable, and second is known as constant region. Like there are two regions. We can say there are two regions. First is variable region and the second is constant region. Okay, let me tell you which one is this. We consider that, okay, basically this region and this region is known as variable of this chain. This region and this region are known as basically variable. The thing which is made up of green is basically variable. This is variable. This is variable. This is variable as well. Variable. Right? Then we have. Okay. Then we have. Let me make the constant region as well. Basically, this region from blue, this region, and this region is also known as basically this is known as constant region. This is known as constant. This is constant region, right? This is a variable region and this is the constant region. Basically, this variable region is known as binding site. This is known as binding site. Binding site. And this constant is basically known as functional site. This is functional site, or it is also known as the site which makes structure. Like it makes it makes structure. So I hope this structure of this antibody is clear to you. Now let's look into the, like, this antibodies are also known as immunoglobulin. Antibody and immunoglobulin both are same thing, right? Basically we have different classes of immunoglobulin, like we have immuno IG, we have IgM, we have IgD. So what is same in that and what is difference? Basically these both have same they have same constant region, or you can say they have same functional site, or their functional site is similar. Their functional site is similar, or you can say that the functional site both are they have same amino acid sequence on the functional site. Okay, but if we talk about the variable site, this variable region, basically the variable region changes with respect to pathogen. This variable region basically changes with respect to pathogen. For different pathogen, there is a different amino acid sequence, right? For one antibody, in one antibody, two antigen can attach. Why? Because there are two binding sites. There are two binding site, but that binding site, like two antigens, must be complementary to each other, or you can say that antigens must be of one type, like one and from in one antibody only two antigen can attach, and the variable site changes with respect to pathogen because the amino acid sequence is complementary to the antigen, right? Now let's talk about the antibodies. Okay, basically like when these antibodies are made, we know that the plasma clone cells, there are plasma clone cells, they make these antibodies. So first these antibodies are attached to them, like in the time of birth of these antibodies, they are first attached to them, and after that they enter into the bloodstream and live. After that they enter the bloodstream and live. Basically we talk about the mechanism of the antibodies. Basically these antibodies combine the toxic substance and neutralize them. That these antibodies combine with toxic substances and neutralize their effect. It basically neutralize their effect, these antibodies. Moreover, we can say that these antibodies basically they activate complementary system. Like we have studied the complementary protein system, so they also activate complement system. And like consider this is the antibody, and this is basically the, you can say, any cell. So this is now become self non self antibody complex. Like basically when this cell attaches with this antibody, it gives signal. This gives signal that macrophages should come and to kill this cell. If two macrophages to kill this cell, right? So I hope this antibodies structure and all the function is clear to you.